So what we found was that IV DNase-1 dramatically improved the anti-tumor activity of the CAR T cells, reducing metastatic tumor burden as well as extending survival.
Welcome back, and thank you for joining us for another Virtual Investor What This Means segment. Today we are featuring Xenetic Biosciences, and I am very pleased to be joined by Dr. Reid Bissonnette. He is Executive Consultant for Translational Research and Development at Xenetic. Thanks for joining us today, Reid.
Thanks, Janine. Happy to be here.
Before we get started, I just want to remind our audience that Xenetic Biosciences is publicly listed on NASDAQ and trades under the ticker XBIO. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's latest SEC filings on their website at xeneticbio.com for the latest information. Reid, congratulations on your recent presentation of this interesting poster at SITC. So investors are familiar with CAR T cell therapies and the success that these revolutionary therapies have had in hematological malignancies. Can you provide for investors a quick background and your thoughts on why we have not seen the same level of success with CAR T in solid tumors? And then what is Xenetic's strategy to overcome these challenges?
Okay, so there are several reasons why CAR T therapies haven't had much of an impact in solid tumor cancers like melanoma, non-small cell lung cancer, colorectal, hepatocellular, and others. First, there's a problem of access. Unlike blood cancers, solid tumors are dense. They have abnormal vasculature, meaning their blood vessels are a mess, and they express factors, cytokines, and chemokines that prevent CAR T cells from effectively penetrating the tumor. So, for example, the extracellular matrix in many solid tumors acts as a physical barrier, limiting immune cell and CAR T cell access. A good example of this is pancreatic or PDAC, which has a hard-to-penetrate physical barrier called the desmoplastic stroma that hampers access to many therapies, including even small molecule chemotherapies. What's relevant to this work are NETs, which also can prevent immune cells from accessing and engaging tumor cells. One reason to treat with DNase-1 is to address this by resolving the NETs. To access the tumor immune cells like CD8 killer T cells, which comprise much of the CAR T cells infused, need to leave the blood and infiltrate the tumor. So the second issue is infiltration from the level of the blood vessels. There are several steps in this process called diaperesis, and several of these are inhibited or suppressed actively by the presence of NETs. Third, solid tumors often present in an environment that is frankly hostile to immune cells, and by extension to CAR T cells. It's immunosuppressive due to the presence of regulatory T cells called Tregs, myeloid-derived suppressor cells, tumor-associated macrophages, and specifically pertinent to this work, tumor-associated neutrophils, which can directly suppress the CAR T cells or, by releasing NETs, contribute to much of the mechanisms responsible for blocking CAR T cell function. NETs can induce also the expression of immune checkpoint molecules. You're familiar with these, PD-1, PD-L1, and others that can directly inhibit CAR T cell activity and anti-tumor efficacy. NETs can induce and be induced by TGF-beta, which is a major cytokine driver of both immunosuppression as well as tumor progression and metastasis. And lastly, CAR T cell efficacy is limited by exhaustion and poor persistence. By exhaustion, I mean cells are chronically stimulated. So exhaustion relates to the immune checkpoints, and persistence is also tied to that, but also to the fact that solid tumors have both antigen heterogeneity and antigen loss. Solid tumors often have variable antigen expression, meaning that CAR T cells, which normally target just one antigen, may not recognize all the tumor cells. And then, due to selective pressure from the immune system, these tumors can downregulate those one or two antigens and shed them, leading to resistance. This antigen heterogeneity is a major distinguishing factor between solid and liquid tumors, though it's worth mentioning that oncologists are also starting to deal with the same issue with liquid tumors. So these four issues, infiltration and trafficking, immunosuppression, antigen heterogeneity and loss, and finally CAR T cell exhaustion and lack of persistence, are all tied in some ways to NETs. Our hypothesis is that we can use DNase-1 to reduce or eliminate NETs, and by doing so, enhance the efficacy of other therapeutic modalities like chemo and immune checkpoint blockade and CAR T to close the gap between the responders and the non-responders.
Okay, so that is very heavy science. Can you summarize what was presented on the poster?
Okay, so the poster presented some work that was done with one of our collaborators at Scripps Research. We set up a model of metastatic melanoma that forms lung metastases in mice. These mice were syngeneic to the tumor, which was B16F10, meaning that they have an intact functioning immune system, and the tumor is the same genetic type as the mouse. B16 is a highly aggressive and very treatment-resistant tumor, so it sets a particularly high bar for any kind of therapy tested, including immunotherapy and CAR T therapy. So first we made the CAR T cells. These were mouse T cells that were inserted with the gene necessary to express the CAR receptor that recognized the mutant form of the epidermal growth factor, EGFR, that is something that's often mutated in solid tumors. We tested the CAR to make sure that they, one, killed the tumor cells and not normal cells, two, and then released factors, namely cytokines and chemokines, that are known to support an anti-tumor response and are used often to as a quality control measure to test the responsiveness of CAR T cells. Then we combined the CAR T therapy with systemic IV-infused DNase-1. We also looked at knocking in the DNase-1 gene into tumor-bearing hosts via infection with an adenovirus that transferred the DNase-1 to the host liver cells, essentially creating a DNase-1 factory. So we used two different ways to deliver the DNase-1, IV infusion and then gene transfection. What we found was that the IV DNase-1 dramatically improved the anti-tumor activity of the CAR T cells, ending up with reduced tumor burden and metastases and extending survival. When we performed the mechanistic studies to try to understand what the DNase-1 was adding to the mix, beyond the obvious hydrolyzing of nets, so what we found was that IV DNase-1 dramatically improved the anti-tumor activity of the CAR T cells, reducing metastatic tumor burden as well as extending survival.
Reid, that is really exciting. So what does all this mean, and what are the next steps for Xenetic with this program?
Okay, so further work here is needed, but we really like what we saw from this study. We also did a pharmacodynamic analysis of the tumor microenvironment after the combination therapy, and what we saw was an increase in the proportion of tumor-infiltrating T helper cells, T killer cells from the CAR T populations, as well as non-CAR T cells from the host, which I should mention is a key finding, since the current belief is that for CAR T cells to be truly effective in treating solid tumors, they must interact with, engage, and even recruit the host's immune system to really be effective. So that's what we observed in these animals. Ultimately, this is first and foremost a significant opportunity to potentially enhance the clinical activity of CAR T cells, anybody's CAR T cells, by adding systemic IV DNase-1 to the treatment paradigm. It's also a significant stepping stone to what we've been planning and have started to work on, which is to develop a CAR T product that brings the DNase-1 along with it. This is called the DNase-1 armored CAR T. So the patient would get the CAR T infusion, and those cells would have the CAR for the specific target on the tumor cells. And at the same time, these cells would secrete DNase-1 locally, where the CAR T cells meet the tumor.
Excellent, Reid. Thanks for that. And with that, this concludes the Virtual Investor What This Means segment featuring Xenetic Biosciences. Thank you, Reid, for joining us today. And if you want to learn more about Xenetic, I encourage you to visit Xenetic.com for more information on the company and to follow their social channels to stay current on the latest information. And for more on Virtual Investor segments, visit virtualinvestorco.com. I'd like to thank everyone and wish you a great rest of your day.